So first, I just want to thank DeBoer as well as the IHHR and everyone else that helped to put this seminar together. I feel like it's a great opportunity for everybody to continue to stay connected but practice their social distancing, especially since most of these conferences this summer have been canceled. So like DeBoer said, I'm Tim Luongo. I'm currently a postdoc in Joe Bauer's lab at the University of Penn. And today I'm going to talk about NAD metabolism and its role in the mitochondria and the heart, and more specifically, its role in heart failure. Also, I included my Twitter handle here. So if anybody has any questions after my talk, or if you are watching the recording and have any questions, feel free to reach out to me. So it's really difficult to begin and talk about NAD without really discussing aging. The aging field really drove the NAD field to where it is today. So for hundreds, if not thousands of years, people have been trying to find this fountain of youth. What it began as is what could have been like a mythological drink or a mystical bath that is represented here in a painting from the 14th century. And it led to many explorations and even the European discovery of Florida. Although the mystical bath or drink never came to fruition, over the last few decades, the scientific community has made significant advances in understanding the mechanisms of aging and actually revealing some of the pathways that can improve lifespan. One of the key features of aging is that actually, as shown here from multiple studies, is that NAD actually declines as we age. And it's thought that this could be one of the causal mechanisms for aging. And it really accelerated the NAD field to where it is today. One of the first mechanisms that was shown to be able to slow aging or increase lifespan is caloric restriction. Here's a study conducted in 1986 where mice underwent caloric restriction, where they reduced their calorie intake by 25 to 65%. And as you increased your caloric restriction, you actually increased the lifespan in the mice. And this has been observed in other animal models, including non-human primates. Unfortunately, if you're like me, and I believe most humans are, food is really life. And so caloric restriction is too much of a sacrifice. And I think I'd rather choose the fountain of bacon over increasing my lifespan. However, that might not have to occur because in 2008, the Garanti lab found one of the mechanisms for increasing lifespan. What they found was that under caloric restriction, NAD levels seem to increase in multiple tissues. And here we're showing skeletal muscle NAD content increasing with caloric restriction. And they proposed that this caloric restriction actually led to an increase in NAD content and activated sirtuins, which activates the deacetylase activity of sirtuins, which had already previously been shown to increase lifespan, which leads to that increased longevity of life. So NAD is an essential molecule that's involved with multiple cellular processes, and these include DNA repair, calcium signaling, inflammation, cellular metabolism, gene transcription and translation, as well as circadian rhythm. But what exactly is NAD and how does it contribute to these processes? Well, it's a molecule that acts as both a coenzyme and a co-substrate. It acts as a coenzyme for energy metabolism by accepting electrons during glycolysis or beta oxidation TCA cycle to form NADH. NADH can then donate its electrons to complex 1 of the electron transport chain to drive that membrane potential and lead to ATP production. Along with being a coenzyme, it acts as a co-substrate. And this is where NAD is actually consumed by NAD-consuming enzymes. And it's degraded to nicotinamide. And it's done so by enzymes known as PARPs, which are involved with ADP ribosylation, or also known as pyrylation. It's a post-translational modification that can be involved with DNA repair as well as cell death signaling. CD38, where NAD is actually consumed to form cyclic ADP ribose, which is a calcium agonist, and then sirtuins, which are deacetylases. And during aging and many different diseases, it's thought that NAD decreases, and this reduced NAD leads to reduced ATP generation, hyperacetylation of proteins, which is thought to be detrimental, reduced DNA repair, as well as cell death. So NAD is synthesized from multiple precursors, but the primary pathway for NAD synthesis in most cells is through the salvage pathway. So here, NAD is synthesized from nicotinamide through the rate-limiting enzyme Nampt, and then nicotinamide mononucleotide is converted to NAD by NMATs. And then it can be consumed and to reform nicotinamide. And this is a cyclical process. NAD can also be synthesized from tryptophan through a de novo pathway, as well as niacin through the Price-Handler pathway. And finally, it can be synthesized from nicotinamide riboside, which is an NAD precursor that has really led to a renaissance in the field. Supplementation of NMN or NR can bypass the rate-limiting step for NAD synthesis and boost NAD just by supplementation. So it might be hard to see this list, but it's really just to show you the number of studies since 2018 that used either NMN, as shown here, for treatment of preclinical diseases, or here where we're showing NR. And all these studies have shown that there's therapeutic potential for supplementing NAD for the treatment of various diseases, and many of these have led to clinical trials. Here I'm just showing a few of the findings. In this study, under high-fat diet conditions, NMN supplementation actually led to improved glucose homeostasis, and here they're showing it through glucose tolerance test. NR supplementation in models of Alzheimer's disease can improve cognition. And there was a science paper in 2016 that showed that NAD can actually extend lifespan through NR supplementation. So since NAD is a critical molecule and its supplementation has shown therapeutic promise, this led the cardiovascular field to wonder what is its role in the heart, and is it relevant in heart failure? The heart is a highly metabolic organ, and it requires kilograms of ATP each day to be day in and day out. And to provide this ATP, the cardiomyocyte's volume is actually composed of 20 to 40% mitochondria. And the primary fuel of the heart under normal conditions is fatty acids. However, in the damaged heart, there is a shift. The failing heart is metabolically dysfunction, there's impaired mitochondria, and the failing heart shifts its primary fuel from fatty acids to glucose and ketones. NAD is an essential molecule to study in heart failure, because in heart failure, NAD is required for all of these metabolic pathways. To assess NAD's role in cardiac injury as well as heart failure, the Shutashima lab investigated what happens to the rate-limiting enzyme Nampt during either cardiac injury through ischemia reperfusion injury or pressure overload-induced heart failure by transverse aortic constriction. And what they found was that the protein expression of Nampt actually decreased. The Merkitske lab also observed a similar effect in human heart failure biopsies when they collect them from patients with dilated cardiomyopathy. And this suggests that NAD synthesis is decreased during cardiac injury and failure. Supporting this reduced synthesis, the Kelly lab showed that they observed a 30% decrease in NAD levels in the left ventricle during heart failure when they looked at human heart failure biopsies from dilated cardiomyopathy patients. Along with a decrease in NAD, the TN lab, along with the Kelly lab, showed that there's a significant increase in mitochondrial acetylation in both models of heart failure as well as in human heart failure biopsies. And it's thought that this increased mitochondrial acetylation causes mitochondrial dysfunction. And the proposed model was that this increased acetylation is due to a decrease in NAD, which decreases sirtuin activity and increases the acetylation. So to combat the loss of NAMPT during cardiac injury, the Shadashima lab either generated a cardiac-specific NAMPT overexpression mouse model, or they supplemented mice with NMN to boost NAD levels. And when they did this, they showed that there was a significant protection of the myocardium against cardiac injury. Since then, multiple labs, including the Tien lab, the Merkitske lab, as well as the Hershey lab, along with others, that NAD precursor supplementations with NMN or NR can protect against multiple preclinical forms of heart failure, whether it's pressure overload-induced heart failure, dilated cardiomyopathy, or even mitochondrial-induced myopathies. And these studies really exemplify the therapeutic potential of NAD precursor supplementation in heart failure. And currently, there are ongoing clinical trials to evaluate its efficacy. So the question in our lab was, does NAD depletion actually cause heart failure? To test this, we generated a cardiomyocyte-specific tamoxifen-inducible NAMPT knockout mice. And just as a reminder, NAMPT is the rate-limiting step for NAD synthesis in the heart. And the reason we utilize a tamoxifen-inducible model is we observed when we constitutively deleted NAMPT in the heart, it was embryonic lethal. So we wanted to reduce the compensation, and we also wanted to reduce the compensation that could occur during development. So we deleted NAMPT in the adult heart. And we did so by delivering tamoxifen for 5 days. We then confirmed NAMPT deletion by Western blot. And our thought was that this would lead to NAD depletion in the heart, and we could determine what happens both metabolically to the myocardium as well as physiologically. So here I'm just showing that the model actually works. So 4 weeks post-tamoxifen delivery or deletion, we see a 70% reduction in the NAD content in the heart. Looking just at the NAD metabolome overall by LCMS, we see that there's a trend for an increase in the tryptophan metabolites when we delete NAMPT. And all the metabolites involved with the salvage pathway are significantly decreased, including nicotinamide, NMN, NAD, and NADH. NADP and NADPH are preserved, as well as methylnicotinamide. So next, we wanted to see what happens both early post-NAMPT deletion. So we assessed at 2 weeks post-NAMPT deletion, and then later, 8 weeks post-NAMPT deletion. So when we measured NAD at 2 weeks post-deletion or tamoxifen delivery, we observed a 60% reduction in NAD content. We also measured NAD at 8 weeks, and it was actually surprising at this point that the mice were able to actually live this long, because the NAD levels were much lower and there was approximately a 90% reduction in NAD. When we evaluated the cardiac function by echocardiography, at 2 weeks post-deletion, we observed a significant reduction in the cardiac contractility, as shown by percent fractional shortening of the left ventricle. And then when we evaluated at 8 weeks, to our surprise, where NADs levels are dramatically decreased, we actually observed a comparable, if not slightly improved cardiac function. Looking at heart rate to body weight as a marker for cardiac hypertrophy, we saw at 2 weeks and at 8 weeks that there was a significant increase in hypertrophy. Since there was a milder phenotype than we expected, we then wanted to test if NAD depletion actually increased the susceptibility to stress. So the first stress we did was more of a physiological stress. We looked at the running endurance of these mice by running them on treadmills at 2 weeks, and we really didn't see a significant difference in performance. And we saw a similar result at 8 weeks, where they had the same amount of endurance when you deleted NAMP compared to the control. To add a pathological stress, we subjected our mice to chronic adrenergic stimulation by implanting osmotic mini pumps containing isobutyrenol. And when we did this, we measured no change in the heart rate in our NAMP knockouts compared to our control after isobutyrenol stimulation. When we assessed cardiac function by echocardiography, we saw that there was reduced contractility 14 days after isobutyrenol stimulation in our controls, but where our NAMP knockouts already had a worse function, the isobutyrenol stimulation didn't decrease the contractility with this model. This suggests that NAD depletion does not increase the susceptibility to chronic adrenergic stress. Since heart failure is associated with mitochondrial dysfunction and NAD plays a critical role in mitochondrial respiration, we wanted to determine if NAD depletion actually disrupts mitochondrial function. So first we wanted to look at mitochondrial NAD pool. And after NAMP deletion, when we did this looking at 2 weeks post-deletion, if you look at the whole cell, we see a 60% reduction, but there isn't a significant decrease in our mitochondrial pool. When we assessed the mitochondrial pool at 8 weeks post-deletion, we saw a 50% reduction in mitochondrial pool, while there's only 10% NAD left in the whole cell pool. This unexpected result, this result was actually unexpected because our lab previously reported that the skeletal muscle specific NAMP knockout mouse had its whole cell NAD and mitochondrial NAD depleted at a similar rate. So this could be a unique compensatory feature of cardiac mitochondria. To measure mitochondrial function, we isolated ventricular mitochondria and then measured their oxygen consumption rate using an Ouroboros high resolution respirometer. At 2 weeks post-deletion, when we looked at the oxygen consumption rate, whether stimulated with pyruvate malate, succinate, or palmitate malate, we saw no significant change in the respiration rate. And just as a reminder though, the NAD pool was actually intact at this time point. At 8 weeks, when we looked at the oxygen consumption rate, we were surprised that even though the mitochondrial pool was depleted by 50% at this time point, the mitochondria actually had increased respiration with these substrates. To see if a decrease in mitochondrial NAD could lead to an increase in acetylation, we evaluated the mitochondrial acetylation state at 8 weeks post-deletion. And when we did this, we observed only a modest increase in acetylation, suggesting that NAD depletion alone cannot increase acetylation. Since we saw an increase in mitochondrial function, we also wanted to assess what happens to mitochondrial complex expression. And when we looked at this, we didn't really see a significant change when we compared our knockouts to our control. Next, we wanted to measure the whole metabolomic profile of the heart at the point where there's only 10% residual NAD. And when we did this, we saw that there was a significant buildup of glycolytic metabolites. However, there was no significant change in TCA cycle metabolites. And this suggests that NAD depletion in the cytosol could be disrupting glycolysis, but it's not really affecting mitochondria. Since our mice were able to live so long without NAMPT expression, NAD levels were preserved for weeks, we wanted to see how the residual NAD was maintained. So what we did was we conducted an experiment where we infused mice with isotopically labeled NAD precursors, nicotinamide, niacin, and tryptophan all at the same time for 8 hours. And then we isolated the hearts as well as the cardiac mitochondria and determined the NAD labeling pattern of these isotopes by LCMS. When looking at the cardiac NAD labeling pattern, the first thing we notice is that all three precursors can synthesize NAD in the heart, but there's definitely a preference for nicotinamide. And then when we're looking at the knockouts labeling pattern, when you're comparing the ion counts to the labeling, you can see that there's a significant decrease in the amount of labeling in the knockout, and it suggests that there's decreased NAD turnover or decreased NAD consumption as NAD synthesis decreases. Another observation we made was that when we compared the percent niacin labeled NAD metabolites to the knockout and the control, the knockout actually had a significant increase in the niacin labeled NMN, NAD, and NADP. Looking at the mitochondrial pool, when we measured the NAD in the mitochondria, it appeared it was only being synthesized directly from nicotinamide. And again, its turnover rate significantly decreased, suggesting that the consumption is also down in the mitochondria to preserve its pool. So since we originally thought generating this mouse model that there would be significant mortality due to metabolic dysfunction and loss of NAD, we wanted to see how long these Nampt knockout mice could survive. Looking at the survival of these mice, there seems to be a two-phase death that occurs. The first is within the first week post-tamoxifen, and then after that 8-week time point, there seems to be this dramatic or sudden phase of death, and by 10 weeks post-tamoxifen delivery, we see a 95% mortality rate. Finally, we wanted to determine if our mice could be rescued from NAD precursors. To assess which precursor is preferred by the heart, we isolated adult cardiomyocytes and then treated them with equal molar ratios of nicotinamide, NMN, or NR for 3 hours. And we observed that cardiomyocytes seem to prefer NR over other precursors. This wasn't surprising since NRK2, the kinase required for NAD synthesis, is one of the most highly expressed proteins in the heart. And it was also shown that by the Merkitske lab that NRK2 expression actually increases in heart failure. We also saw that its expression significantly goes up with loss of Nampt. Unfortunately, serum levels of NR are very low, so it can't completely compensate for the loss of Nampt to maintain NAD levels. For our experiment, we wanted to see if oral NR supplementation could rescue our Nampt knockout mice. So what we did was we did an experiment where, and we did this experiment because our previous data suggested with isotopic tracers that oral delivery of NR is mostly broken down to nicotinamide. So in our mouse model, we hypothesized that oral delivery of NR could not synthesize NAD due to the loss of Nampt. However, when we measured NAD levels after delivering 500 milligrams per kilogram of NR for 13 days, we actually saw restoration in the NAD content in our Nampt knockout hearts. When we measured cardiac function by echocardiography, we saw a significant improvement in cardiac function as measured by percent fractional shortening after delivery of NR. And finally, NR prevented the lethality that was observed in our Nampt knockout mice. So just to summarize my results, cardiac-specific NAD depletion causes cardiac dysfunction and hypertrophy. Decreased cardiac NAD does not reduce endurance or susceptibility to adrenergic stress. Cardiac mitochondrial NAD pool is protected from depletion. Mitochondria with reduced NAD actually have normal or enhanced function and only modest increases in acetylation. And decreasing NAD appears to alter glycolytic but not TCA cycle metabolites. Finally, oral NR can rescue NAMP knockout hearts from NAD depletion. So just to overall summarize, it appears that a modest depletion of NAD that is observed in heart failure cannot actually cause heart failure. In our model, we're actually decreasing NAD much more by 70 to 90%. And at this point, we're only seeing moderate cardiac dysfunction, almost normal mitochondrial function. And it's not until you get less than 10% that you start to see this lethality or heart failure to begin. So first, I just want to thank all of you for listening to my webinar today. And I also want to thank those who were involved in this project, especially my mentor, Dr. Brower, for all his support. Carthy helped with the infusion of the mice with the isotopes, as well as we've got some guidance from the Kelly lab, as well as the Penn Cardiovascular Phenotyping Core. And we have a great collaboration with the Rabinowitz lab, and Lynn Wang and Melanie helped with looking at the metabolome as well as the isotope labeling. And Marie helped with the synthesis of the isotopes. And I also have to thank all the funding agencies that supported this project. And now I'll open up for any questions.
Well, thank you. Thank you very much. This was really actually fantastic. Let me just put us both on the gallery view, and hopefully if you can maybe stop sharing, Tim, maybe that would be perfect. I mean, amazing. I think there's going to be lots of questions, and I think
yeah,
I am right actually, because they're already starting. So you're gonna, well, it's up to you, Tim, how long you want to spend responding to these.
Okay.
So, and I would also like to just let everyone know that Tim's kindly agreed for this webinar to be recorded. So it will be put up on the YouTube channel probably this evening, the latest tomorrow morning. And yes, I will just now let Tim answer the questions. Maybe if you could just read out the name and the question, that might be useful, because not everyone will be able to see them.
All right, the first question is from Matthias, and his question is, it's so surprising to see that NAD at the low levels has such a low impact. He asked how we were extracting the NAD. So we're extracting the NAD by either perchloric acid extraction or with a methanol extraction for LCMS. So we confirmed our levels with both procedures. And yeah, we're in the chloric acid, it'll definitely extract from all the cellular pools. So Benjamin asked, did we look at isovolumetric relaxation or mitochondrial performance index? And no, we haven't looked at any of that yet. What is the relative expression of NAMPT in cardiomyocytes compared to other cells? NAMPT, I'm not really sure if it's enriched in the heart, but it definitely is pretty ubiquitously expressed throughout all tissues. Another question is, what is the life expectancy in these knockouts while the NAD content is low? So I showed that with a 10% NAD left, once you get below that threshold, you start to see that the NAD pool, or you start to see that increased mortality as the NAD pool goes down.
So Matthias,
was the heart rate measured in conscious, Mike? No, they were actually under anesthesia at that point. So Jonathan asked, did we look at FAD levels? Is there any compensation? And actually in some of our steady state metabolomes, we actually see that FAD is decreased as well. did you try something that's more mitochondrial targeted like doxorubicin? No, we haven't tried any other stressors than what I've shown right now. And what's the dose that we used in the isobutanol pumps? We used 75 mg per kg, so it's quite a high dose.
That's Sam Dudley there.
Yeah. Samuel, did we measure mitochondrial ROS? No, we haven't measured mitochondrial ROS yet. Charles. So Charles asked about the LV diameter. And one thing that's surprising is that we're not really seeing a dilated cardiomyopathy like you would expect in this model. So it's more when the mice actually die, we see it looks like more of a hypertrophic cardiomyopathy. And this spontaneously occurs because we're not really seeing it with echocardiography. It's right as they're dying. Someone asked or said that Samuel asked said that low NAD is associated with arrhythmias. We haven't measured the ECG on these mice, unconscious mice, so we're not sure if it's causing arrhythmias. So Michael asked, can you take NR and still increase your bacon consumption? And, you know, that's still a huge question in the field. Is this supplementation actually going to work in humans? And there's been some clinical trials so far that have shown that there's only a very mild effect when supplementing with NR. So we're not really sure why that's occurring and why the mouse studies aren't translating. Obviously, this is a common effect, but I can't say that you can take NR and instead of doing caloric restriction to increase your lifespan yet in humans.
So you wouldn't supplement your bacon with the NR?
No, I don't think so. We haven't looked, this is a question from Julia. We haven't looked to see if NR supplementation can rescue our lifespan. I actually just did the NR experiment right before we got sent and locked into our houses. So that's why it's a weird time point of 13 days. So I had to sack the mice right before pen locked down. So we haven't been able to extend that study any further. We haven't, Matthias asked, have we tried any more severe cardiac stress? Like I said, the only stress so far we've done is the isobutanol stress. And we thought since it's such a metabolic stress that would have pulled out a phenotype, but we could try something like TAC to see if that increased susceptibility.
Yeah, another similar question to Sam's from Dan Johnson.
No, yeah, we plan on doing telemetry. We just haven't done it yet. So Timothy asks about the mitochondrial acetylation, and this recently has come into controversy because Deb Muo came out with a paper showing that you can hyperacetylate the mitochondria and you still don't see a dysfunction. So we're not really sure, but it looks like just depleting NAD by 50% doesn't increase acetylation. So it's not really driving acetylation. So someone asked if we saw a difference in beta oxidation metabolites. So we did see a decrease in our knockouts in the palmitoylcarnitine levels. So it is possible that there's increased beta oxidation in our mice at that point. And someone was asking what the cause of death at 10 weeks was post-tamoxifen, and we think it could be based off our data, just looking at the steady state metabolism, is glycolytic failure, not actually mitochondrial failure. And that would be through GAPDH. So Fabio asked, how is the NADP pool maintained when NAD content is decreased? Well, our lab has shown in studies that the NADP pool has a much, much slower turnover rate than the NAD pool. And I also showed that it appears that the niacin can feed the NAD pool without Nampt. Another question about supplementing, seeing if it protected against death. We haven't done that yet. Sam asks, have we put our mice on high-fat diet? No, everything you saw is pretty much what we have so far. It's a completely unpublished study, and it's still ongoing.
So are you writing this up, or have you submitted it yet, or?
So we're writing it up now since we have time and can't really work. Yeah, and Rosanna asked what would be the main cause of death. Again, we're leaning towards it's probably glycolytic failure. Either you have one of your dehydrogenases in the cytosol we think is failing. So were there changes in the cardioprotective genes or proteins? That's something we haven't really seen. John Elrod, he's my PhD mentor.
So you got to answer well.
So what's the hypothesis for the differential regulation in the mitochondrial pool? Could there be an alteration in transport? Well, this is something that we're studying extensively in the lab, and we've identified how NAD is transported in the mitochondria. But in this case, it's a combination effect. It's possible that NAD transport is increased in these mitochondria as NAD depletes, and we've seen this in other stress conditions, like in the liver as well as in the kidney under hemorrhagic shock. We see that even though the whole cell pool decreases, that the mitochondrial pool actually increases. So it could be some type of stress response. So it's possible that there's an alteration in transport, but along with that, we're seeing that there's a decrease in the turnover rate, suggesting that there's decreased consumption, probably by sirtuins. The upstream NAD depletion in heart failure, that would probably be through Nampt. Like I showed, the Shachishima lab showed that Nampt expression is decreased. And then another question is, what is the consumption pathway for NAD in the mitochondria? And that would be sirtuins, especially SIR3. Yeah, so he's mentioning how our mice phenocopy Nampt overexpression, which develop cardiac hypertrophy. And this is tricky because in that model, they vastly overexpress Nampt to very high levels. So it's a little hard to say that just boosting NAD alone can cause hypertrophy because it could be just dependent on Nampt expression. We haven't looked at NAD's association or effect on NOx, nor have we looked at the methionine salvage pathway in our Nampt knockout mice. I've already addressed the mortality. So in our study, both sexes are included. So this was a question from Benjamin. Is there a sex difference? And we really don't see a sex difference. And this really points me to the opinion that it's probably not a mitochondrial effect, because a lot of times female mice are protected from mitochondrial associated diseases. And in our mice, we see a similar phenotype, whether it's male or female. So we didn't see a change in ATP, but we didn't look at the creatine phosphate levels in our mice. So we can't really comment on that. So the only comment that I can make for Swapnil's question, what happens to in the human heart to tissue NAD content during heart failure, is that it appears to decrease. And then Matthias's question. So Matthias's question is asking if oral NR bypasses gastrointestinal as well as the hepatic barrier that were proposed by our lab, that NR is broken down to nicotinamide. And we're really not sure, because in this study we haven't used a isotopic labeled NR to see if NR is actually fully making it to the heart. But we have seen that oral NR in our skeletal muscle Nampt knockout, that very small amounts of NR can make it there as a complete molecule orally. But we still have to investigate it further. It still is possible that NR is being broken down to nicotinamide and then being taken up by a neighboring cell, say a fibroblast, and then converted to NMN or NR, and then going into the cardiomyocytes. Since this is a long-term study, we still have to investigate that further with labeled isotopes. So we did RNA-seq, and we didn't see changes in SOD levels, but we haven't checked the protein expression. And the concentration of our NAD precursor in the 3-hour cardiomyocyte experiment was 1 millimolar. And someone asked about histone acetylation. So overall, the acetylation state, if you look at the whole cardiac lysate, there's not much of a change. So we're not really seeing a huge change, but we haven't looked specifically at histones. But one thing that was surprising when we looked at the RNA-seq, there was very little change in the transcriptional levels of all genes. So it didn't appear that NAD had a huge role in changing transcription in the heart. I think that's all the questions.
Yeah, that was Emma. Emma Robinson asked that. So I think there's one more question actually in the chat from Swapnil Shivale. And actually, don't know if you can see that, but it's on what happens in human heart failure to tissue NAD content.
Like neighboring tissue? Yeah, we haven't, in human heart failure, no one's looked at the other tissue metabolome yet that I know of. So we're not sure if the liver levels decrease or anything.
Right. I mean, there's actually, so weirdly enough, there's questions coming in a Q&A still. So there's one from Kozubie. I don't know if you can see it, but I'm just seeing them pop up.
Mine ends at Emma.
So yes, but it seems to be filling in earlier on for whatever reason. So he says, what was your NAD precursor concentration in your 3-hour cardiomyocyte experiment?
Oh yeah, I already answered that. It was 1 millimolar.
Oh, sorry, I apologize.
That's okay.
So I think, I mean, there's you've gone through 40 odd questions. So, so well done. I'm quite impressed. I think you've definitely deserved your Easter chocolates now. So,
Thank you.
so I mean, thank you also to everyone who's tuned in. And as I said, thankfully, Tim has agreed for this to be put up on a YouTube channel. And also for any of the other talks, you can register and using the Google Docs spreadsheet, which I hope you'll have available. If not, there is always a follow-up Zoom email where the link to the Google Docs spreadsheet and the YouTube channel are available. So, I mean, Tim, I'd like to thank you.
Thank you. It was great.
And also, I'd like to thank the ISHR again for helping us run these series as well. And hopefully everyone can get their chocolates for the rest of the day. And we join again with Professor Roger Fu from National University of Singapore at the same time.
Thank you.
So thanks, Tim. Enjoy the rest of the day.
You too.
Thank you. Bye-bye.